with the new topic, which is of your second semester, and it is in your medical microbiology paper. So the topic or the unit number is 4, and the name of the unit is chemotherapy. So it is the most applied part of our day-to-day life, because chemotherapy is the way which is used to treat the different infectious diseases and so as to kill the infectious agents. And whenever there is any infection, whenever there is any disease, we treat ourselves by the prescription which is given by the medical practitioner, the physician. So the physician always diagnose the disease and accordingly gives us the treatment. And that treatment is nothing but, the scientific term for it is chemotherapy. Many times this chemotherapy is misunderstood. That is, chemotherapy means the treatment which is used for the cancer treatment. No doubt, for cancer treatment, this chemotherapy word is used. But scientifically, chemotherapy is the application of chemical drug for treatment, means for therapy. So the world is made up of two parts. Yes, I have started the lecture. Are you listening me? Can you listen me? Can you see my slides? Siddhi Shinde. Siddhi Shinde, unmute yourself. Yes, Sakshi.
Ma'am.
Yes, can you listen me?
Yes, ma'am. But
And
slide is not visible.
Yes?
Slide is not visible.
Okay, so when you will communicate to me, okay, it's all right. If again I will try to share the slide. If you could not see, please call, let me know. Okay, is it visible now?
Yes, ma'am.
Okay, so this chemotherapy, though we say it's chemotherapy, the commonly term which is used for the treatment of cancer patients, actually it is not only for the term is not only related to the cancer. So chemotherapy is use of chemicals. It is chemo and therapy. So chemicals, the chemical drugs, they are used for the therapy, means treatment. So use of chemicals for the treatment, use of drugs for the treatment is the simple meaning of chemotherapy. So whenever there is any disease and we visit a physician, according to the diagnosis, the physician gives us a regime. Regime means doses of a specific drug and how they are to be taken. So that is doses. Not only the drug is prescribed, but the doses also are prescribed. That you have to take the single tablet in a one day, or you have to take 2 tablets in a day, or whether you have to take 3 tablets in a day, or whatever may be the case. So that is the dose allotment, and that is known as regime. So such type of the regime is given for the patients suffering from different diseases. And these chemical drugs which are used for the treatment, and that all regime totally is known as chemotherapy. This chemotherapy is defined as use of chemical substances in treatment of infectious diseases or diseases caused by proliferation of malignant cells so as to destroy infectious agent or malignant cells without damaging host tissues. Now this definition has 3 different parts. The first part is it is use of chemical substances. Then where to use it? In treatment of infectious diseases or diseases caused by proliferation of malignant cells. That is second part. So There are not the diseases which are caused by the infectious agents only. Means the bacterial diseases, viral diseases, protozoal diseases, and so many infectious organisms they cause the disease. And their treatment only is not the chemotherapy, because many a times there is no any causative agent, there is no any disease, but the disease is caused, and that is due to proliferation of malignant cells, that we call it as cancer. So it is use of chemical substances in the treatment of infectious diseases as well as in treatment of the cancers. For what purpose? So as to destroy infectious agent or malignant cells. It is to destroy the infectious agent or malignant cell. So we are using the chemical substance for the treatment of infectious diseases or diseases caused by proliferation of malignant cells. And third part, why to use? It is to destroy the infectious agent or malignant cells. And the last and the most important, when we are using these chemicals so as to kill the malignant cells, so as to stop the proliferation of malignant cells, so as to kill the infectious agent, it should not harm the host. So without damaging the host tissue, without damaging the host cells, the others which are pathogens, those are to be killed by use of chemicals, and that process is known as chemotherapy. So chemotherapy is use of chemical substances in treatment of infectious diseases or diseases caused by proliferation of malignant cells, so as to destroy infectious agent or malignant cells without damaging the host tissue. So that is the central idea or definition of the term chemotherapy. So who discovered this type of the technique, that is use of chemicals for the treatment of infectious agents or to inhibit the proliferation of the malignant cells? And the credit goes to the person who is known as father of chemotherapy, Paul Ehrlich. If you do remember, we have studied the contributions of Paul Ehrlich in BSc first. So he is the first person who used chemicals for the treatment of different diseases. So the first chemical he used was arsefinamide. So that is the chemical which was marketed. The trade name of this trade name of the arsefinamide is salvarsan. Now, what is the trade name and what is the chemical? So when we say paracetamol, or that paracetamol is the single component. So actually paracetamol is the trade name, and the chemical component in that tablet is its the constituent. So if you are having for example, which I can give you is the common example of Rabidi. Suppose Rabidi is the name. So what is the constituent or component of that drug? So that is some antacid. So such type of the different names are there. One is the chemical constituent name and another is market name, or the name by which that drug is marketed. That name is known as trade name. Cefaxime. Cefaxime is the name which is written on the strip of the tablet, but their constituents are different. So those constituents are the chemicals, and the name which is printed on the strip of the tablet, that is its trade name. So there are different types of such trade names which are given, and we purchase the drug by the trade name, but its constituents are different. So this arsefinamide is a chemical constituent which was marketed as by the trade name salvarsan, and it was for the treatment of syphilis. So syphilis is the disease. We know it is a sexually transmitted disease caused by a spirochete, Treponema pallidum. So Paul Ehrlich was the person who for the first time used arsefinamide chemical for the treatment of this syphilis, which is caused by a spirochete, Treponema pallidum. At that time, quinines were there, and those quinines were used for the cure of malaria. Nowadays also we commonly use quinine for the cure of malaria. Also, these arsenical compounds were used to cure trypanosomiasis. Trypanosoma gambians is the causative agent, and this trypanosomiasis causes the disease which are related with the mental disorders, or which are related with the disorders which are related to sleep, not exactly the mental, but these are related with the sleep disorders. So many times such type of the organisms were killed at that time also by the use of chemicals. It indicates this certain malaria is caused by a protozoa. Salvarsan is used for spirochete. So at that time, chemical agents acting on spirochetes and the protozoa were available. So at the time of Paul Ehrlich, that is the near about 19th century. So it was the beginning of 19th century, and in the 19th century first few years, the chemicals were used to treat the diseases which were caused by spirochetes and protozoa. But there was no any chemical available for the treatment of diseases caused by bacteria. So bacterial diseases were not treated by use of chemotherapy at that time. And so that was a major question, because there are so many different types of the bacterial diseases, and those diseases were not treated by using any of the chemical. But it was later on found that 1935, Domag was the first man who for the first time used sulfonamide called protonsil for successful treatment of streptococcal infections. So in the time of Paul Ehrlich, the treatment were available for spirochete infections. They were also available for protozoal infections, but were not available for bacteria. Streptococci are the bacteria, and these streptococcal infections were for the first time treated by the scientist Domag in 1935. So what he used for this treatment, and those are nothing but the sulfur compounds known as sulfonamides. So the sulfonamides, and which were marketed as a protonsil, were used for the successful treatment of streptococcal infection. And this discovery were given the Nobel Prize to Domag. So for this discovery, Domag was awarded with the Nobel Prize, that is use of the sulfonamide for the treatment of bacteria, the first chemical used for bacterial infections. So later on, or simultaneously, there was discovery of the penicillin, but the penicillin which was discovered by Alexander Fleming was not purified, and so there were not such type of the research done. But in 1940, Flory Chain and his associate produced a stable concentrated form of penicillin, and from that development, research was concentrated to find antibacterial compounds from microbial origin, and those are known as antibiotics. Up to that, the chemicals were synthesized, and in the factory, all the chemicals were designed, all the chemicals were prepared, and were treated or were used for the treatment of diseases. But the discovery of the penicillin changed the scenario. Instead of synthesizing chemicals in the factory, they used microbial factory, and they started search for such microbial factory so as to produce the antibacterial compounds. So penicillin was the first antibacterial compound which was produced by a bacteria and was acting against other bacteria, and such type of the chemical known as antibiotic. And from that discovery, and from the purification and concentration of the penicillin, from 1940, the chemicals were searched for. There was a research, and it was about the antibacterial substances which can be obtained from the microorganisms and are known as the antibiotics. So these antibiotics, so it may be any agent. It may be a agent which is chemically synthesized. It may be a chemical agent which is originated from a microorganism, whatever it is. So any type of such chemical agent can be used for the treatment of the diseases, because always it is not possible to have the antibiotic. Many times we have to use chemical agents still today. So in chemotherapy, when the chemotherapy is applied, we will have to think of what are the general principles when we apply the chemotherapy. So in chemotherapy, it is a total effect. Chemotherapy is a total effect of what? It is total effect of chemical agent, pathogen and host. So what is that effect? The effect of chemical agent is on infectious organism and host to cure disease and improve hygiene. So chemical agent kills the organism and improves the hygiene of the host. So this thus the chemotherapy becomes an interaction between the drug, host and pathogen. So it is an interaction. So if you see this triangle, all the three are interacting with each other. So no one is separate. We can't think of only host, we can't think of only drug or only pathogen independently. So in chemotherapy, all these factors are interacting with each other. So this interaction has its own characteristics. Own characteristics, and these three factors has their own properties. So this interaction is nothing but the chemotherapy. So this drug should act on host, the drug will act on pathogen, or drug will act on pathogen, and as the pathogen is killed, the health of the host. Yes.
स्लाइड नाही दिसत.
आधीची दिसली का बाळा?
हो आधीची दिसते आता आधीची दिसत नाही.
एकदम ती गेली का? बर, पहा दिसतीये का?
हो
दिसली ना? ठीक आहे. तर so the drug will act upon pathogen will be killed, and so the health of her host will be improved, and such type of interaction is nothing but the chemotherapy. So in this chemotherapy, these three factors has their own properties. They don't interact with each other, they are having their own characteristics or own properties. So what are those own properties? So properties of the drug. So the first component of this chemotherapy of the triangle is drug. So the important property of the drug is this drug may be chemically, it may be synthetic, it may be semi-synthetic, or it may be natural. So natural drug means the drug which is obtained from directly plant source or microbial source. So nowadays we know that there are different Ayurveda's drugs, and these drugs are nowadays recommended. Even the in COVID situation, if we remember the advertisement made by Ramdev Baba, Coronil. I will not go in the details whether it is really a effective drug or non-effective drug, but his claim is that it is totally obtained from the natural vegetation of the plant sources. So that is natural drug. Or in our Ayurveda, there are different drugs. For example, the drug like Adulsa Sarbat. So that is, or Kumari Asav. These are all the natural drugs available because they are obtained from the plant sources and by the process of fermentation, etc. So there is no any chemical synthesis. So whatever is naturally present, or stevia, if you remember about the diabetic drugs or the sugar which is used for the diabetic patients, so that is natural source. So such type of the natural sources which are used for the preparation of the drug, those are called as natural drugs. So these different types of the drugs, even tulsi, ginger, even clove, cinnamon, these are all the sources. Now these are all considered as the drugs. So these are the sources of natural drug. Then synthetic drugs, like as I told you, sulfonamide. Sulfonamide is totally synthetic drug. But semi-synthetic, yes, semi-synthetic means partly which is obtained from the natural source, and partly it is modified by some synthetic or some chemical process. The best example is different types of the penicillins which are now available. Because we know that the penicillin is obtained from the Penicillium chrysogenum or Penicillium notatum, means the fungus penicillin, sorry, penicillium. So this penicillium fungus produces penicillin. So this penicillin has three structural parts. The first part is the thiazole ring, second one is beta-lactam ring, and third one is the side chain. So what happens in the semi-synthetic penicillins? These two rings are kept as it is. So the natural penicillin, that is penicillin G, is produced by the fungus penicillium. But while the penicillium is growing to produce penicillin, the variable part of this penicillin molecule is the side chain. And this side chain depends upon which type of chemical precursor is added in the fermentation media. So if in the fermentation media you change the precursor, the side chain structure changes, and depending upon the side chain structure, the type of penicillin also varies. So you have studied the penicillin fermentation. So you know the types of penicillin and how this penicillin X, penicillin V, penicillin G, etc. So penicillin G is natural penicillin, but penicillin X, penicillin V, these are semi-synthetic because their side chains are synthesized by addition of a required precursor, and so they are known as semi-synthetic. So this drug may be natural, may be semi-synthetic, or may be synthetic. So any drug, it must have a broad spectrum even in low concentration. So it broad spectrum means what? What is spectrum? Spectrum means range. So this drug should act upon a variety of microorganisms. So this drug should be acting upon bacteria. It should be acting upon gram-positive organisms, gram-negative organism, or it should act upon protozoa. It should act upon different types. It should inhibit at least few protozoa. So whatever it is, but it have broad spectrum, means it can kill, it can act upon different varieties of microorganisms. That is known as broad spectrum. So whatever the drug you are using or you are selecting for the treatment, so this why these properties of the drug we have to study, because depending upon the properties of the drug, we will have to, or the physician has to select the drug for the treatment. So while selecting the drug, these properties are considered. So the drug must be broad spectrum. Then second one, the agent, the drug must be lethal to pathogen. So it should be killing only the causative agent. So when it is applied, the agent should be lethal to pathogens only, and it should not affect, or it should not have any bad effect on the host. Many times what happens, the drug which is taken shows many side effects. So what are these side effects? Side effects indicate that drug is acting on the host cells also. So many times this host is getting affected due to pathogens, and so the property of the drug should be, it should only be lethal to pathogen and it should not affect the host. Another thing, it should be effective at body temperature. Very simple thing. So if drug has to act, then this drug, we are giving this drug to the host. And so either it may be taken orally or it may be injectable. So how drugs are given? Mainly the tablets are oral tablets, or certain injections are given depending upon the case of the patient, depending upon the type of drug available. So this will enter into the body. And where this drug has to act? The drug has to act in the body. And what is the body temperature? 37. So this drug also must be active in 37 temperature. It should be effective at the body temperature. If the drug is effective only at room temperature, that drug though is given, because what we do is all the study of the drugs is carried out in many times in vitro instead of in vivo. So in vitro means in laboratory conditions. So in laboratory conditions, the drug though is kept at the exact 37 degrees Celsius. When the drug is applied, where if the person is suffering from fever, what will be the body temperature? Whether it will be 37? No. So whatever is the body temperature, suppose you are applying paracetamol, you will not give the paracetamol to the person who is having normal body temperature. The paracetamol will be given to the person who is at higher body temperature. So when you have to give the drug, that drug should be active at that body temperature. Or suppose there is a person having diabetes and you are giving the drug for diabetes or for sugar control, and if that drug is acting at the temperature of 39 degrees Celsius, then and it is ineffective at 37 degrees Celsius, whether that drug will be able to control the sugar? No, because the normal person, though the person is having higher levels of the sugar in their blood, their body temperature is normal. It is 37. So that drug should act at 37 temperature. So thus, depending upon the conditions, different types of the drug, we have to, or the person has to say or to see that whether that drug is effective at the specific body temperature. So that is also the property of the drug. Then it should not alter upon standing. So this is very interesting. So the drug should not alter. Means what? The drug is having some antimicrobial property, or the drug is have certain anti-allergic property. Whatever is the property of that drug, whatever is the activity of the component of the drug, that should not decrease after certain time period. So if the drug is kept for a longer time, then also its activity should be maintained as it is. So it should not alter upon standing. Not only its activity should be decreased, but even there should be no any change in the activity. So originally the organism, originally the drug was acting upon some some pathogen, and after few days that drug changed the property, and now it is not acting on that specific pathogen, but is acting on the normal flora of the body. That is changed activity. So such type of the changes there should not happen when we select a drug. But the most important thing is it should not lose its activity upon standing. So my friends, if you go now, actually you are in the home, as soon as I finish my lecture, you have to see any drug from your home and see the label, what is written on that. So that should not alter upon standing. So this is the property which is always mentioned on the drugs. And but the name or the sentence which is required to show this property is little bit different. So what is it? Can anybody guess? The drug should not alter upon standing. Yes, there is a specific period given. This study is also done in the pharmaceutical laboratory, that after how much days, after how much years, the drug is losing its properties. And accordingly, there is printed dash dash that you can use this drug before that period. So that indicates that drug is altering its properties after that specific period. So what is the specific term which is printed on the bottles of the drugs or a tablet strip? Can anybody tell me?
Expiry date.
Very good. Who answered the question?
Manali.
Very good, Manali. So that is expiry date. So there should not be any alteration upon standing. So we use a drug before expiry date. So after expiry, if you use that drug might have changed its property. It might have changed to the toxicity. There may be toxicity increased, or there should be ineffective components of which are the important components of the drug. So that is also the property that the drug should not alter upon standing. Then another property, it should not produce tolerance in the pathogen. So what is that tolerance? So whenever we use any drug, and if we use the drug repeatedly, after few years, after a certain time period, the pathogen get acquainted with that antibiotic or that drug. It may be an antibiotic, it may be a chemical drug. So repeated use of a same drug will lead to produce a tolerance, means acclimatization or adjustment of that pathogen to the drug. So if we use the same drug for many times, pathogen now knows about the drug and pathogen changes itself so that it gets adjusted to that chemical, it gets adjusted to that antibiotic. So naturally what will happen? That pathogen will not be killed. Now it is adjusting. Many times what happens, pathogens use that drug after certain exposure as a food material. They changes themselves. Pathogens are very clever. They never will surrender the situation. They never will try to die. They always will try to survive in whatever is the adverse situation. So drug availability in the surrounding of the pathogen is also the adverse environment. So for few generations, pathogens will die, no doubt. But after certain generations, the pathogen will change its morphology or its certain physiology so that that drug environment now will not be adverse to that pathogen, but it will be favorable. And pathogen will use that drug many times. It is also observed as a nutrient, and that drug will be used as nutrient by the pathogen. So pathogen, instead of getting killed, gets grown and it multiplies and its number goes on increasing, and so the disease also goes on increasing, goes to the severe cases or serious cases. So such type of the tolerance should not be produced by the drug. So the drug should be so powerful that pathogens should not overcome its activity. So the drug should have a property that it will not produce tolerance, resistance power, that is tolerance. Tolerance means resistance in the pathogen. It should have a uniform composition. How drug should be? Drug should be in uniform composition. So this is mainly applied for the liquid drugs. Those are mainly the syrups. So if you also observe any liquid bottle of the drug, the drug bottle in which there is a liquid syrup or liquid drug, what is written on that bottle? Can you tell me? It is related with the uniform composition of the drug. What it is written? Shake well before use. So there is a sentence written, shake well before use. Now this also you have to check. As you have to check the expiry date, you will have to check this uniform composition instruction. Why it is written? Because when the drug or when any liquid drug is kept as it is for a period of 2 hours, 3 hours, if it is not having solution form, if it is in the suspension form, the suspended drug settles down. So in many cases of the drugs which are mainly used for the kids, always there is a bottle. In that bottle there is powder, and on the top portion of the bottle there is marking, and it is written there, or the doctor gives us the instruction that boil the water, cool the water, and fill the water in the bottle up to the mark. They mix the powder and the water thoroughly so that all the powder gets dissolved in the water. And once it gets dissolved in the water, then give 5 ml to the baby. So such type of the dosage are prescribed by the doctor. Now what happens? This powder which is there and we add the water, many times this powder do not get 100% dissolved in the water, and some particles, some powders remain suspended in the liquid, in the water. So when suppose you give the first dose, you keep the bottle in the rack or in the cupboard, and after 5 hours or 6 hours you have to give the second dose. Now within these 6 hours, what can happen? That this powder will settle down, and at the top you will get only the colored water. And in the second dose, if you give without shaking, if you give directly the dose to the kid or the baby, you will give only the colored water, and the actual active component of the drug, that is powder, that will settle, that will remain at the bottom of the bottle. Naturally, what will happen? The kid, the baby, will not get the sufficient amount or quantity of the drug, but will get only the colored water, and there will be no use of that dose. And the baby, you will find that is not curing. Why is not curing? Because you are not given the equal or correct amount of the drug to the baby. Why it happened? Because you didn't shook the bottle properly before giving the dose. So always remember, whenever there is any liquid drug, you will have to always shake before use. Never forget, shake before use. So thoroughly shake it, immediately take out the required amount, quantity, and give it to the baby. Keep it as it is. For the next dose, when we will get the bottle out, again you will have to shake well. So then and then you can get the drug in the uniform composition. And why this happened? Because the drug was not completely soluble in the water. And so the next property of the drug is the drug should be completely soluble in the water and other solvents. But other solvents mean which? For example, alcohols. But many times the drugs which are given orally are mainly used for the solubility, or they are search for solubility in the water. So the drug should be soluble in the water. It should penetrate the host cell. Whenever the drug is given to the host, naturally it should enter into the host cell, because so many pathogens are intracellular pathogens. If the pathogens are outside the cell, if they are in intracellular spaces, then no doubt they will come in contact with the drug. But if the drugs are if the pathogen is intracellular, to reach to the pathogen, the drug must enter, the drug must penetrate the host cell. So the property that drugs must penetrate the host cells. Because if the drug do not come in contact with the pathogen, it will not affect the pathogen. So there should be always a direct contact of the drug and the pathogen. Then another property, it should not affect the natural defense mechanism of the human host. We know that the natural defense mechanism is the best defense mechanism against any infectious organism. So the natural defense is like phagocytosis, production of antibody, etc. So this is the natural defense mechanism of the human host. If the drugs are affecting the natural defense mechanism, the person will become more and more susceptible to other infections. So if you are killing a single pathogen and the defense mechanism is lowering down of the person may get other infections also. So the natural defense must be as it is, or it should be always higher. And if the natural defense is proper, actually there is not infection. Whenever the natural defense lowers down, at that time there is always an infection. And this natural defense, when is lowered, there is infection, and we take the drugs to kill the pathogen and not to affect the natural defense mechanism. But always it is observed that the defense should come over the pathogen. The natural defense should increase, and this is our property of the body. Whenever there is infection, immediately the natural defense, like primaries, first line of the defense mechanism is taking place. But if the pathogen enters into the blood, then there is requirement of the production of antibodies or other types of the defense chemicals. But it takes certain time. Immediately as the pathogen enters, immediately there is no production of the antibody. And up to that the defense mechanism is again properly reaching to the level, up to that time there should be the antibody, there should be the killing of the pathogen, and that killing of the pathogen will require the drug, but that drug should not interfere with the natural defense mechanism. Otherwise, there will be never production of the antibody, and we will have to be rely upon only the antibiotics. So there should not be any effect on natural defense. The drug should not be inactivated by stomach acids and blood proteins. So as I told you, how the drugs are given, basically there are two ways, like oral and another is injectable. So when we take orally the drugs enter into the stomach. Stomach is having acidic pH, stomach is having different types of the juices and acids. Even we inject drug, the drug is injected directly in the blood. So the blood proteins may interfere with the activity of the drug, or when orally the drug is taken, in the stomach, whatever the stomach juice is there, which is comprising acid and other enzymes, these also may act upon the drug. So there should not be inactivation of drug by the stomach acids or blood proteins, otherwise drug is of no use. And the most important thing, the drug should be available easily and it should be cheap. Nowadays, we know there are certain diseases of which the treatment is very, very costly. Generally, the genetic diseases, the diseases which are genetic disorders, in that genetic disorders, the treatment is very, very costly. The drugs also are not available. In many conditions, we have to bring the drugs from the foreign countries because our country is not having the stock of that drugs. And when such type of the drug we have to bring from outside the country, that is always very much costly. As there is no availability, it is not cheap, and such drugs cannot be afforded by many people in the country. So, generic drug system, whatever is nowadays popular, it is for the availability of the cheaper drugs. So, the drugs must be cheap and available, and that is done by the generic drugs. So, remaining in the next lecture. Yes. So, remaining we will see in the tomorrow's lecture. Okay. If you are having any query, if you are having any problem, if you don't understand any point, just you let me know in tomorrow's lecture. I will clear it. Okay. Thank you.